Hey everybody, welcome back to another video. Today we will discuss the role of serotonin, also known as 5-hydroxytryptamine or 5-HT, in modulating the excitability of neurons in the dorsal cochlear nucleus, and how this sometimes can become unbalanced, potentially contributing to tinnitus. Now, the studies that I'm going to be mentioning here are work by Tang and Trussell, 2015, Felix et al., 2016, and Thompson et al., 2001. All of these studies will be linked in the description if you want to do some further reading. Now, before we dive in depth into the role of serotonin, or 5-HT, in dorsal cochlear nucleus physiology and how imbalances can contribute to tinnitus, it is super important to understand why serotonin receptors are in the DCN at all. Now, a lot of people might think that the DCN is just a relay for auditory signals from the cochlea. But as we know from my previous videos, it is actually a multisensory integration hub that receives a lot of inputs from different sources. These, of course, include projections from somatosensory pathways and also the serotonergic inputs, which we will be discussing today, arising mostly from the dorsal and median raphe nuclei. Now, what these are clusters of serotogenic neurons near the midline in the brainstem. Now, the serotonin receptors are in the DCN for a few reasons. One of them being is, of course, the fine-tuning of auditory processing. The DCN processes complex acoustic cues, such as vertical sound localization cues based on spectral notches. And serotonin can dynamically modulate the excitability of fusiform cells in the DCN, for example, to either enhance or suppress responses depending on behavioral context, like arousal, attention, or stress. Now, if we're talking about multisensory integration, specifically somatosensory information from the SP5 pathways or other ones, I'm talking about the trigeminal nerve and the dorsal column nuclei, serotonin can actually shape these non-auditory influences. And basically, the presence of 5-HT receptors provide a mechanism to adjust how the DCN combines this auditory and somatosensory processing under different states, like sleep-wake cycles, vigilance, for example. So basically, by having 5-HT receptors, the DCN can be, quote-unquote, turned up or down in response to changing levels of mood-related stuff like being aroused or just, general mood like, fury or anything like that, which actually is very, very important in filtering or amplifying these signals, especially in challenging environments or under stress. So imagine a person 20,000 years ago walking around in the jungle and he's being hunted by a tiger, for example. The serotonin receptors obviously would be activated very intensely due to this person's extreme vigilance and stress in the current situation. And this would actually amplify the signals that are being processed by his dorsal cochlear nucleus to obviously respond more to very, very slight sounds, for example, that he might consider or that his brain might consider to be dangerous, like the tiger making a noise in the bushes, for example. And this is very useful for survival. Now, under typical conditions, the balanced activation of serotonin receptors does help maintain sort of an equilibrium between excitatory and inhibitory circuits in the dorsal cochlear nucleus, or the fusiform cells and the Cartwheel cells. And this balance does help avoid hyperactivity that can manifest as tinnitus or something else like hyperacusis. And if this balance is misregulated, like a person taking antidepressants, it can contribute to maladaptive plasticity and potentially the neural noise in the auditory pathway that can already be dysregulated by hearing loss, for example, can be amplified even further. And such filtering systems like the thalamus or the medial geniculate body can interpret this as actual noise instead of blocking it out like it's supposed to before passing it to the auditory cortex to be perceived as noise. In other words, tinnitus. Now, moving forwards, we're going to be talking about paths of serotonin innervation within the DCN and neighboring regions. We're also going to be talking about 5-HT receptor subtypes, specifically 5-HT2A/2C, 5-HT7, and others that can either excite or inhibit DCN neurons by altering their membrane currents. We're also going to be mentioning the mechanisms through which antidepressants, for example, SSRIs, might destabilize the delicate excitatory-inhibitory balance, or the EI balance in the DCN, sometimes either causing or worsening tinnitus. We're also going to talk a little bit more about thalamic gating, specifically the thalamus, or more specifically the medial geniculate body. How it can gate abnormal signals traveling from the DCN to the auditory cortex. Now, a brief anatomical overview. Now, based on those studies that will be linked in the description, they have shown that the serotonergic axons project really heavily from the dorsal and median raphe, which are both located in the midbrain, near the midline, to multiple cochlear nucleus subdivisions, including the dorsal cochlear nucleus. And these raphe neurons release the serotonin, or 5-HT, from their terminals, which form varicose fiber segments in the DCN. If anybody's wondering what I mean by varicose here, I think everybody knows what varicose veins are. And what I meant is, or what varicose refers to here, are the naturally occurring bead-like enlargements, which can be seen along the axons or fibers of the neurons. Now, they have a very high density in the molecular layer of the DCN. In other words, the most superficial layer, which contains the apical dendrites of the fusiform cells and Cartwheel cells, plus the parallel fibers from granule cells. If anybody is wondering what apical dendrites are, we all know that dendrites are the projections of a neuron that receive input signals from other neurons, and the word apical means the apex or the top of a structure. And apical dendrites typically extend upwards from the tip of the neuron's cell body, or the soma, toward the outer layers, which allows the neuron to receive multiple inputs from distant sources. And this is how fusiform cells, for example, receive input from the parallel fibers of granule cells, or the somatosensory pathway. Now, basically, because the dorsal and median raphe nuclei are functionally diverse centers, their extremely widespread innervation in the DCN means that the levels of serotonin, or 5-HT, can shift rapidly in response to changes in stress or arousal, including those that are mediated by internal states or external stimuli. Now, this divergent projection patterns allow for a single raphe neuron, or just small amounts of them, to co-modulate huge clusters or large parts of the DCN. Now, to get into the real complex stuff, in vivo studies suggest that fusiform cells express 5-HT2A/2C and 5-HT7 receptors. And activation of these receptors triggers downstream signaling via G proteins, cAMP, and SRC kinase pathways, which enhances the HCN channels, also known as hyperpolarization-activated cyclic nucleotide-gated channels. And these channels mediate IH, which is a depolarizing inward current when the membrane potential is hyperpolarized. Now, about these HCN channels and action potentials. HCN channels are activated by hyperpolarization, or when the cell becomes more negative or below the resting potential. And these channels open and allow the influx of sodium and a small amount of potassium ions, which generate the inward current, or the IH that I mentioned. And this inward current helps bring the membrane potential back towards a resting or threshold levels. Now, this is extremely important in neurons with rhythmic or pacemaker activity. In other words, for example, cardiac pacemaker cells or neurons in the thalamus. Now, back to the activation of the 5-HT2A/2C and 5-HT7 receptors. As I mentioned before, the activation of these receptors enhance the HCN channels, which then leads to the resting membrane potential becoming more depolarized or less negative, and the threshold for triggering spikes or action potentials lowers, which increases the overall firing probability. And in many neurons, the first spike latency shortens, and spike jitter, or the variability in spike timing, decreases. And overall, these effects collectively boost fusiform cell excitability, leading to an increased chance of responding to subthreshold or near threshold sounds. A bit more about Cartwheel cells. As we know, Cartwheel cells are inhibitory interneurons in the molecular layer of the DCN, and they have complex spikes and provide glycinergic and possibly GABAergic input onto fusiform cells. Now, typically, Cartwheel cell activation suppresses the fusiform cell firing, and if serotonin either does not strongly excite Cartwheel cells or it differentially excites fusiform cells more, an imbalance can occur. And in some experiments, 5-HT can actually increase Cartwheel cell firing by depolarizing them, which then boosts the inhibitory input to fusiform cells. In other cases, 5-HT might excite the fusiform cells more. Now, the net effect depends on the receptor subtypes, which are expressed between Cartwheel cells and their relative distribution versus them in the fusiform cells. And this complexity means that small changes in 5-HT receptor expression or function on Cartwheel cells versus fusiform cells can drastically alter excitatory and inhibitory balance. And when that balance shifts, the DCN's output either becomes more hyperexcitable or more suppressed. And in the context of tinnitus, the hyperexcitable scenario, where, the fusiform cells become more active than inhibited, this is, of course, very problematic. Now, if we're talking about vertical cells or other inhibitory circuits, the vertical cells in the deep layer, they also provide powerful inhibitory inputs, especially to fusiform cells. And if 5-HT or serotonin upregulates vertical cells significantly, that might reduce fusiform cell firing. On the other hand, if fusiform cells are more sensitive to 5-HT than vertical cells, the fusiform excitation could dominate. But basically, again, the pattern of excitatory and inhibitory receptor distribution does dictate whether the DCN's excitability, or the net, or the overall excitability, goes up or goes down in response to serotonin. Now, again, about excitatory-inhibitory balance in tinnitus. In normal physiological conditions, or mild arousal or moderate stress, serotonin modulates DCN circuits in a manner that matches environmental or internal demands, and auditory signals are filtered appropriately, with the cartwheel and vertical cells providing the correct the timely inhibition to fusiform cells. And then the 5-HT sets the gain at a moderate level. Now, if we're talking about hyperexcitability, or the pathological state, the excess 5-HT or excess serotonin can drive fusiform cells to fire spontaneously or respond too much to minimal inputs due to the lowered thresholds. And it also can be reduced inhibitory effects if cartwheel or vertical cells are underactivated, or if their synapses are weakened, for example. This can exacerbate the fusiform cell hyperactivity. And over time, fusiform cells can develop abnormal spontaneous firing patterns that propagate along the ascending auditory pathways, possibly passing through or failing to be gated by the thalamus, leading to the tinnitus perception. So in general, an imbalance where excitatory drive outweighs the inhibitory checks, or vice versa in fewer cases, can prime the DCN for persistent spontaneous activity. Now, about antidepressants, SSRIs, and tinnitus. Now, selective serotonin reuptake inhibitors, or SSRIs, they function by blocking the reuptake of 5-HT into presynaptic terminals, which then prolongs and increases its presence in the synaptic cleft. Now, while SSRIs are extremely common for depression and anxiety treatment, they can raise 5-HT levels significantly in many brain areas, including the DCN. And in patients who develop or who spike or worsen their tinnitus after starting SSRIs, one possible explanation is that the chronic elevated 5-HT drives the fusiform cells to a hyperexcitable state, or disrupts the normal inhibitory circuits, the cartwheel and vertical cells. Instead of having the beneficial fine-tuning effect, the net effect is a more robust and consistent depolarization of fusiform cells, less reliable or insufficient inhibition, and increased spontaneous spiking that is basically tinnitus. Now, some SSRIs, or the time frame of their usage, could lead to downstream plastic changes. In other words, receptor downregulation that might eventually reduce the net excitatory effect on the fusiform cells, or the DCN neurons overall. This could explain why not all people on SSRIs experience tinnitus, or why some patients can actually see some improvement over extended usage. But in certain susceptible individuals, like people with tinnitus, the short-term or long-term changes can actually make tinnitus worse. Of course, as we all know, every person is different. Every person has a different level of somatosensory influence, or the maybe heightened excitatory inputs from the SP5 pathways due to, cervical issues, tension, jaw issues. So it's very tough to say how exactly SSRIs will affect certain individuals. Now, some interesting facts on cartwheel cells. Firstly, Cartwheel cells have a special firing pattern, also known as complex spikes, which, interestingly enough, are similar to the firing pattern seen in Purkinje cells, which are neurons found in the cerebellum. The similarity in their firing pattern is linked to the fact that Cartwheel cells and Purkinje cells share similar shapes and functions. And they resemble how circuits in the cerebellum operate. And as we all know, they provide inhibition onto fusiform cells, which shapes their output responses. And they are also subject to parallel fiber input from the granule cells. Now, the granule cells themselves can be influenced by serotonin because 5-HT in the DCN often arrives from the parallel fibers in the molecular layer. And, cool fact, if Cartwheel cells become hyperactive under certain 5-HT receptor dynamics, fusiform cells can potentially be suppressed. On the other hand, an insufficient Cartwheel activation leads to greater fusiform cell excitability, or more tinnitus. So potentially, researching some medications that affects the 5-HT receptor dynamics in such a way that causes the Cartwheel cells to become more active could potentially suppress tinnitus. But 5-HT and serotonin dynamics are very complex, and finding or researching some medication that will affect exclusively Cartwheel cells, either being inputted from the parallel fibers or whatever, it's very complex, and having a med be so selective is very, very difficult. But basically, serotonin's potential enhancement or suppression of granule cell activity can potentially also shift how the DCN processes somatosensory signals, which of course complicates the interplay of excitatory and inhibitory circuits. I think that recent research shows that gene therapy approaches using viral vectors might be able to selectively alter the activity of Cartwheel cells in the dorsal cochlear nucleus to help suppress tinnitus. And again, Cartwheel cells are the inhibitory interneurons that release the neurotransmitters like glycine and GABA, and they normally keep the DCN's fusiform cells in check or from becoming overactive. Now, using viral vectors, we could introduce genes into the Cartwheel cells targeted specifically for the Cartwheel cells that either enhance their inhibitory output or make them more responsive to certain drugs or signals, which could restore a healthy balance between excitation and inhibition. I think that boosting the natural suppression provided by Cartwheel cells is the key to reducing tinnitus. As we all know, dysfunctional ion channels and stuff like that is responsible for this disruption of excitatory and inhibitory balance. And actually, when using the Susan Shore device, I didn't really see any immediate effects until a few days post-usage, if we're talking about tinnitus specifically. Now, if we're talking about the ventral cochlear nucleus or loudness hyperacusis, I did see an immediate effect post-usage. But as we all know, the ventral cochlear nucleus, which according to Susan Shore's research, is responsible, or at least partially responsible, the Bushy cells for loudness hyperacusis. It diverges from the dorsal cochlear nucleus. So having my hyperacusis and tinnitus change in different ways or different manners post Susan Shore device usage makes sense. And the reason why I think my tinnitus continued changing post-Susan Shore device usage was that something happened with my Cartwheel cells in the dorsal cochlear nucleus, which eventually provided inhibition towards the fusiform cells. So I think that the Susan Shore device, it obviously does have a profound effect on the Cartwheel cells as well, due to their somatosensory or SP5 input. And what I think happened in my case was that the hypersynchrony was disrupted with the Cartwheel cells, and they regained some of their functionality, and with time they started providing more adequate inhibitory signaling to my fusiform cells over time. So this means that the Susan Shore device does not necessarily have to provide immediate suppression to tinnitus. The tinnitus can decrease with time, and we're talking days, if not weeks, post usage of the device. Now, there's a lot of things that I would like to say about thalamic gating. And thalamic gating is very important, not only for tinnitus, but also for visual snow syndrome. And research does suggest that HCN channels contribute very significantly to the resting membrane potential. And as we all know, as I mentioned previously, 5-HT has a very profound effect on HCN channel activity. And again, as I mentioned before, neurons with pacemaker activity, like neurons in the thalamus, are very strongly affected by HCN channel activity. Now, more on this later, of course, because visual snow syndrome is a very complex condition that requires a completely separate video in the future, which I will be making eventually with the help of particularly knowledgeable members in my community. But overall, we can see that everything here is related. Everything is related. 5-HT, thalamic gating, HCN channel activity, ion channel dysfunction, all of this, it's related to each other. And potentially, I do think that viral vectors do have a massive potential for modulating this ion channel activity, to restoring it to its normal state. But again, I'll make a separate video on viral vectors in the future. That's about everything I wanted to say today. See ya.